How's it going, everyone? It's Chow here, and today we are going to talk a little bit about Mendelian genetics, so let's get started. Okay, so some important vocabulary to get out of the way. We have the word allele. So allele and gene are often interchangeable, but if we're going for specifics, an allele is it's just a type of gene, so it's a form of a gene. So think about big A, little a, they both represent a gene, but each allele can be one specific form. So big A is an allele, little a is an allele, and big A, little a both form a gene. Yeah, there you go. That works. Yes. So an allele is just a different form of gene, but oftentimes you have alleles and genes being used interchangeably. Here are another two words that are sometimes interchangeable, and that is the character versus the trait. So sometimes we use character, sometimes we use trait, but if we're going for more specifics, a character is an observable physical feature, so for instance, like eye color, whereas a trait is a specific form of a character, for instance, red eyes or blue eyes or brown eyes or something like that. A heritable trait is one that's going to be able to be passed from parent to offspring. So that should be pretty straightforward. It's just a trait that can be inherited. And then we have genotype versus phenotype, which are two very key terms that are used in Mendelian genetics. So genotype is basically the alleles on the chromosome, whereas phenotype is the physical characteristic that's going to be observable. We then have this word over here called true breeding, which are basically just families of organisms or groups of organisms that are able to consistently pass down specific characteristics, or I guess I should say specific traits, from one generation to another. So there you go. They're consistently passing down a certain trait, which makes them true breeding. We then have the breakdown of generations, which include the P generation, which is the parental generation, as well as the F1 generation, which is the first filial generation you get from crossing the P generation. You can also sometimes see this as P1. And then you can actually cross the F1 generation and make an F2 generation or a filial 2 generation. So some basic vocabulary to whet your palates. Now, if we go down to Mendels experiments themselves, obviously we talk a little bit about crossing. But what really Mendel was trying to do, perhaps, at the very beginning was looking at this theory of blended inheritance. So what blended inheritance is this concept that offspring is just the average or mix of the two parents. We know today that's not exactly true to 100%, but at the time there wasn't much information about it. So Mendel used these pea plants, there you go. So Mendel used these pea plants to complete crossbreeding tests and basically do a quantitative as well as qualitative analyses of inheritance. So pea plants have both sort of male and female sex organs, and Mendel or any researcher can basically remove certain sex organs, for instance, the male sex organ, the stamen, in order to control pollination and fertilization and prevent, for instance, self-pollination. And he actually studied about 7 different characteristics. I believe some sources say he did actually a little bit more than that, but there are sort of 7 key characteristics that he looked into. This is more or less trivial information in terms of memorization, but just some basic background that might be interesting. So Mendel was really well known for the monohybrid cross. And what basically a monohybrid cross is just a single characteristic that you're looking at. And he was able to create sort of true breeding plants for each trait. So again, true breeding is basically families of organisms that can consistently pass down a certain trait from one generation to another. So he's able to make these true breeding plants, and he was able to cross those parental true breeding plants to get an F1. And then sometimes you have instances where one trait appeared throughout the entire generation, which eventually is what's known as a dominant trait. And then in other instances, you have basically crosses which resulted in traits that previously disappeared to come back. So when he crossed, for instance, the P1 generation to get the F1, they might only show one specific trait. But when he crossed those F1 generations again, whether it's backcrossing or with each other, some of those traits that previously disappeared actually reappeared again, and supposedly these are what's known as recessive traits. And then through a generalized monohybrid cross, we often see the F2 has this ratio of 3:1 of dominant to recessive phenotypes in the offspring. So this is where that 3:1 ratio comes into, based on specific traits that are being chosen for a monohybrid cross. Which basically gets to sort of our first law here. And Mendel's first law was the law of segregation. And the law of segregation simply states that two alleles of an individual are basically separated and passed on to the next generation singly. So we have this particular gene over here, the big A, little a gene. So the A gene, we should just call it. And the A gene has two different types of alleles. We have big A and little a. And these are going to be passed on independently in the gametes. So that's the first law, or the law of segregation. So in terms of basically crosses, we can use some of these things to help us predict the offspring. And this thing is called the Punnett Square. The Punnett Square is a good way to basically visualize the crosses themselves, at least in monohybrid crosses. Also technically dihybrid, but once you get into more and more characteristics, it gets a lot more complicated. So Punnett squares basically show us the probability of each possible gamete forming, and it can be used to also predict allele combinations, which makes us very, very happy because we can look visually and see these characteristics and these traits being shown. And so you can basically put one parent up here, one parent over here, and you can just simply cross those letters over there and make the correct offspring. So when it comes to sort of these offspring over here, as well as the parents themselves, basically there are kind of two groups if you're looking at sort of a genetic nomenclature type series, and these terms are homozygous and heterozygous. So homozygous are basically alleles on the homologous chromosomes that are the same. So they can be dominant or recessive. If you have big A, or little a, these are going to be basically homozygous alleles. If you want a heterozygous situation, this is basically where alleles on the homologous chromosomes are different. So you have big A, little a. So that's heterozygous, which should make sense both, in terms of scientific reasoning as well as maybe some logical reasoning. So homozygous, big A, little a, or heterozygous, big A, little a. So this should be something that's pretty straightforward and you should definitely have down in your brain. So what if we run into a situation, right, where we don't necessarily know the genotype of the parental generation, for instance, or the genotype of one specific organism? So this is a problem that arises when you have a situation where the alleles are being passed on sort of at an autosomal dominant or autosomal recessive situation. So a particular organism might show a dominant trait, for instance, a purple flower color over here, but you can see that the purple flower color as a phenotype can have two genotypes. It can be big B, or big B, little b. So what if we want to figure out the genotype and we don't know? Well, we can run basically a test cross to see this. So a test cross is used when the genotype cannot be determined from the phenotype. So that particular individual perhaps could be a heterozygous or maybe a homozygous dominant individual, if we're talking about sort of this autosomal dominant recessive type of inheritance. And so what we can do is we can actually cross that unknown individual with a homozygous recessive, and we can actually kind of understand and figure out which genotype that outcome is based on the offspring. So ignore this one over here, but let's look at this particular example over here. So imagine you have your individual, so this particular individual, it could be big T, little t, or big T, big T. So we don't know the genotype of this individual up here. So we cross the unknown up here with a homozygous recessive, which is over here, and we get some offspring from that. And we can see that all of the offspring in this particular example, remember this is just this specific example, it's big T, little t, and we get this particular phenotype. So the big T phenotype is being shown because this is a dominant situation. So again, this is a dominance type of inheritance. Obviously, if it's a different type of inheritance, it might be a little bit different. But let's assume that we know these are the offspring and we get the offspring. So using a particular Punnett square, we can figure out what this particular allele is. And very obviously, it's a big T. So we can use a test cross to basically figure out the genotype of whatever organism we're looking at. But this is, of course, you have to kind of know which type of inheritance the system is following. Is it actually something that's just an autosomal inheritance that's based on dominance and recessiveness, or is it something else? So that's something you have to keep in mind. So then Mendel made it a little bit more complicated, and he did what's known as a dihybrid cross. So instead of looking at one characteristic, we're now looking at two characteristics. And so these dihybrid crosses are very useful because they're able to basically look at whether characteristics are on the same chromosome or different chromosomes, or in a more even broad way, whether those genes are going to be on the same chromosome or whether they're going to be on different chromosomes. So what he did was he crossed two plants, one dominant for two traits and basically one recessive for two traits. And the result was that the F1 generation all showed both dominant traits. That was a really loud motorcycle. I hope that didn't pick up. If it did, whatever. Okay, so the F1 generation basically shows both dominant traits, which kind of makes sense. But what he found out was that if he did an F2 generation result, so you cross the F1 generation with each other to get an F2 generation, it ends up with a 9:3 to 3:1 ratio. So a 9:3 to 3:1 ratio in the phenotype. And we could see it right here. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9 of these smooth yellow ones, or these rounded yellow ones. And we have 1, 2, 3 of these ones that are green and fairly smooth looking. And then we have 1, 2, 3 here that are yellow and kind of rough looking. And then we have basically one over here that is green and sort of rough looking. So this is a 9:3 to 3:1 F2 generation ratio, which is really fascinating because this suggests that there's really not direct connection of one set of genes with another set of genes. So Mendel then proposed his second law, which is the law of independent assortment, which basically states that alleles of different genes sort independently, kind of like so. And the way the chromosomes line up along the metaphase plate kind of influences the resulting daughter cells, whether it's going to be this particular gene is going to be on this side or on this side, and that can influence how it's going to be sorted into the gametes. What's really also important is that assortment in one homologous chromosome occurs independently from assortment in other homologous chromosomes. So basically what you're thinking about here is that one homologous chromosome isn't necessarily affecting another, which means that if the genes are going to be on different chromosomes, they're not going to be connected to one another, and they're going to be basically passed on and assorted independent of one another. Hence the law of independent assortment. We will eventually figure out this is not completely true. So basically, in summary, there are a bunch of different types of allelic interactions that are available. You have ones that are just basically dominants, and then you have ones that are basically codominant. So these interactions are where both phenotypes are kind of expressed in the heterozygous form. You have incomplete dominance, which is kind of a blend of phenotypes in the heterozygous form. And then you have sex-linked, which are basically ones that are carried on the X chromosome. You also have other types of connections such as polymorphisms, pleiotropy, and epistasis, penetrance, and expressivity. And these can kind of mess up the straightforward dominance, recessiveness, relatedness, and inheritance of your normal monohybrid or dihybrid crosses. But generally speaking, these are the main ones that you probably have to kind of focus on. Okay, so one more thing that we have to basically look over is this concept of genetic linkage and recombination. So remember the law of independent assortment, right? Well, genetic linkage can actually mess up that concept, that law of independent assortment. So genetic linkage is basically a situation where genes are on the same chromosome. And this is going to help violate that law of independent assortment because the genes are going to be on the same chromosomes, they're going to be linked, and so you're not really necessarily going to kind of kick them apart, except if you have recombination. So recombination occurs due to crossing over events during prophase 1 of meiosis, and this can actually allow different genes on the same chromosome to basically interact independently and be moved to another homologous chromosome. And that basically allows the chromosomes, those genes, to kind of shuffle around. And even if you have genetic linkage originally where the two genes are found next to one another, by having recombination, you effectively remove that linkage by kicking that gene to another arm of the homologous chromosome next to whatever homologous chromosome it was originally on, if that makes sense. Recombination results can cause differences in the offspring that are not necessarily seen in the parent. So if you get offspring from a cross and you get the parental characteristics show up, so the parental traits are showing up, right? So offspring have same genotypic combination as the parents, then that means you just have a parental effect. But if you want a recombinant, so an offspring that's a recombinant, they basically have new genetic combinations that were not seen in the parents or either of the parents. So if we look at something like this, which is basically recombination frequency, it's basically asking how often does the recombinant occur in this particular offspring group. And so the recombination frequency is simply the number of recombinant offspring divided by the total number of offspring, and that's your RF, or your recombination frequency. The higher the recombination frequency, the further apart the genes are. The smaller the recombination frequency, the closer the genes are together. So if we have an example like right here, we have, let's see, a 17% chance of crossing over. That means that the genes are going to be 17 map units apart, which is what we use to designate that recombination frequency. So if you want to basically look at the correlation, the percent, so not the recombination frequency itself, but the percent recombination frequency, so you have to multiply everything by 100. If you multiply the RF by 100, which gets you your percent recombination frequency, it equals the number of map units those genes are apart, which basically is also equal to the number of centimorgans apart. And so you should definitely know what these terms are. Now, ultimately, what you're able to do potentially with all these things that we learned is basically build kind of a pedigree. So it's a human pedigree is just a genetic map of an entire family for a specific trait that we're looking at. So we can use recessive traits, we can use dominant traits, we could look at sex-linked traits or some other trait that's present in this particular familial group, and basically correlate it all together, build that tree, and see whether it is going to be inherited in a certain way. So you can basically map out different traits that are present in the family and use the presence or absence of those traits in specific family members to see whether that trait is something that's recessive, that's dominant, or sex-linked. And that might be something that you should do for preparation for exams and whatnot, where we can just give you, a situation like this where here's a family human pedigree. What is this particular trait, or how is it inherited based on what you can see? And that might be a good practice question for you to look over in preparation for exams. So I think that's basically all I have for Mendelian genetics. I hope you all found this at least mildly helpful. And as always, best of luck studying, and I will see you in the next video.